Recording in progress.
Okay, now in the chapter number 2, we have to discuss how the organisms are made up of. In the first chapter, we have discussed in detail about the classification, right? How the organisms are classified, how different organisms are classified on this planet, and how we have a separate group for vertebrates and another group for arthropods. We
Yeah.
have done
Right.
all that, right?
Yeah.
Now we are going to discuss particularly how these organisms are made up of. Okay, so as you know, just like in chemistry we have discussed that the smallest thing is, just like in chemistry there was atom. Similarly, when we talk about the living organisms, we say that they all are made up of cells. Okay, they all are made up of cells. Cell are the too small, they are the smallest unit. And when we talk about the cell, we say that simply cell are basic, cells are structural, and functional unit of life. Okay, this is the
Yeah.
basic definition, simple definition of cell. Okay, what is meant by the structural and functional unit of life? It means within a cell there are different structures present which are responsible for life processes. Okay, just like what are the structures present in the cell? For example, there is nucleus, right? There is
Yeah.
mitochondria and
Nucleus.
sorry,
Nucleus.
nucleus, mitochondria, and there are different things, ribosomes as well.
Yeah.
So these are structures within the cell, and these structures are responsible for life processes to take place. Similarly, there are some functional units, and these cells are also functional unit. How it is a functional unit? It performs different activities. Okay, it performs different activities in the body. The cell performs different activities in the body. On this basis, we say this is a wholesome definition of the cell, that cell are structural as well as the functional unit of life. Why is this so? Because there are different structures within the cell which are responsible for different functions. Clear? Are you clear with this definition?
Yes. Yes.
Okay. Now they have mentioned all the living organisms are made up of units called cells. Although cells may take on very specialized functions, you see they have mentioned now. Although cells may take on very specialized function, they have certain common features. This is what they are telling us, is that as we have discussed the classification, there are different classifications of the organisms, so their functions will be different. Just like in the previous chapter we discussed that some organisms, they breathe through gills, they have gills, and some breathe through lungs, right? Some have book
Yes.
lungs, some have gills, some have lungs, just like us. So their function may be different, but some of the common things will be some things will be common in all the organisms. For example, within the animal cell, there will be nucleus. Okay, in all the animals, whatever the category of that animal is, every animal cell will have nucleus. Just like, for example, we have discussed the arthropods, invertebrates. In the vertebrate class, we discussed the birds, mammals, reptiles. They all are different from each other, but there are some things which are common in all of them. Okay, so now we are going to discuss them. These are shown on the opposite page. Both animal and plant have cell surface. Okay, firstly what they are telling us is that there is cell surface membrane, the outer membrane. This is common. The cytoplasm is common, and nucleus.
Yeah. Nucleus.
These three things are common in all the animal cells. Okay, and plant cell. Not only animal cell, but all the plant cells as well. What are the common things? Cell membrane, cytoplasm,
Cytoplasm.
and
Nucleus.
nucleus. Okay.
Also, what about cell wall?
Sorry.
Cell wall. Is it also common?
Cell wall.
Yeah.
That is only in plant cell. That is not in animal cell.
Okay.
Okay, so that's not a common thing. These are the three things which are common in animal and plant, both the cells. Okay, cell membrane, cytoplasm, and nucleus. These three features can be seen on photograph of a liver cell below. This one, they have mentioned this is the liver cell. On this you see this is a nucleus, right? Outside this is a membrane, and this red portion is what? Cytoplasm, right?
Yeah.
So these are the three common things. In addition, plant cell have a now in plant cell, what is the additional thing in the plant cell? A cell wall, right? Cellulose. Cell
Yes,
wall is made up of
cellulose.
cellulose. Yeah, this is not present in the animal. This is only present in the plant cell. A
Plants.
vacuole is also only present in the plant cell, not in animal cell.
Also chloroplasts to make food.
Yes, chloroplast. So these are the three things which are additional in the plant. These are not present in the animal. What are the common things? What are the common organelles?
Cell membrane, cytoplasm, and nucleus.
Yes. And what are the things which are only present in the plant cell?
Cell wall made out of cellulose, vacuole, and chloroplast.
And chloroplast. Yes. Now they mentioned this, a palisade cell from a leaf. This is a leaf cell using a light microscope. In this you see this outer one is the cell membrane. Okay. Over here there is a vacuole. A vacuole is on this side, maybe it's sometimes present. And there is green color containing chloroplast. Clear?
Yeah.
Okay, now the light microscope. You know about the microscope? What is the function of microscope?
Like to what's it called? Magnify something that's like not visible by the naked eye.
Yeah, the microscope is used to see the objects which are too small, which our
Yeah.
eyes cannot perceive. The things just like the atom, we cannot see any atom, right? Similarly, we can't
Yeah.
see cells. You see your hand. In your hand there are fingers. You just see your fingers. Okay, you can't go beyond that. So to see the cells, we need microscope. And microscope, as its name indicates, micro means small, scope means to elaborate. It elaborates the smaller things. Okay. So what happens is that basically from here, if we check from this, how image is viewed, there is a series of lenses over here. Okay, there are lenses present over here, or this position and this position. These lenses help to magnify. Okay, firstly, one lens is there for the focus. It focuses the light. The other lens, it elaborates the picture. It elaborates the cell. Okay, and gives us the elaborated image. So there are different types of lenses. Now they have mentioned cells are too small to see with the naked eye, so microscope is used to study them. Visible light passes through a suitable specimen. Okay, a specimen is the, what you know is sometimes we have a slide glass made up of glass. Okay, just like this, and on this you put in the center you put a drop of the blood, maybe a blood, a drop of the anything in which you want to check the smaller things. Okay, your test sample is known as specimen. Okay, whatever thing you want to check, you want to see the elaborated image, is known as specimen. Okay, are you getting this? Just like for example,
Yeah.
I want to check on a blood drop, that what are the things in the blood. So what I will do, I will take a drop of the blood in the center of this glass plate. Okay,
Yeah.
and this is known as glass slide, and on this slide in the center I have taken the specimen of the blood. So this is known as specimen. They mention visible light passes through a suitable specimen and a series of lenses magnifying the image that is formed. Okay, they magnify the image. A light microscope can give a useful magnification about 400 times. Okay, it means if an object is like of this size, you will see an image of this size. Clear? If the object is of this size, 400 times, which means the image seen is actually 400 times larger than the specimen. Okay, the actual specimen is this, but you will see the elaborated image which will be 400 times magnified as that of original. Okay. The contrast between the different structure in image can be improved by using dyes or strains. Okay, sometimes there are things which are transparent. Okay, so it becomes difficult for you to check what are the things present in those specimens. So what you do, you add some color, you add some dyes on that. Okay, so when you add the dye, that thing prominent. For example, in this specimen is, let's consider this specimen as it is transparent. Okay, it is transparent. Now what you need to do is, you want to check what are the things in this specimen. So what you will do, you will add dyes. Okay, so by
Yeah.
adding those dyes, different colors will appear on it. Those different colors will tell you the different things present in it. Are you getting this? For example,
Yeah.
this is the specimen, this is transparent, I have added a dye on it. Okay, so that dye will prominent, maybe it will prominent the proteins in it. So wherever the proteins will be, those will prominent like this. Okay,
Yeah.
so this is the function of dye. And sometimes we use the dye for the samples which are transparent. Okay, check
Yeah.
the organelles in them, we add the dyes. The nucleus of an animal cell, for example, shows up particularly well when stained with a dye called methylene blue dye. What they have mentioned is that the nucleus of an animal cell, for example, shows up particularly well when stained with a dye called methylene blue. They are telling us that sometimes you cannot see all the things of the nucleus, without a dye. If you want to see all the things of the nucleus, what you need to do, you will add a dye on it. That dye will prominent each and everything of the nucleus. Okay,
Yeah. Yeah.
I am repeating this. They are saying, sometime if you want to check what are the things present in the nucleus, you take the specimen of the nucleus on the glass slide, and you put this glass slide over here, but you cannot check all the things in the nucleus. To check all the things in the nucleus, what you need to do is, you add methylene blue dye. Okay, this dye will prominent all the things present in the nucleus. Okay, and for plant cell, what you need to add, you add iodine solution. Okay, in animal cell you add methylene blue solution, and in the plant cell you add iodine solution. Clear?
Yeah. Yes.
Now can you tell me why there is need of a microscope?
Uh, so like you can see like atoms that what's it called, that like you can't see normally, that are very small.
Yeah, they are very small, you cannot see them. And
Yeah.
so how microscope will help to see them?
It helps by like it like magnifies, like the lens magnifies the image that's gonna form.
Yes, the image will be magnified by 400 times.
400.
Yes, and sometimes we need to use dyes or stains. Why we need to use dyes and stains?
Like to make the color,
Sorry.
like so you can see like the plant or the picture better.
Yes, sometimes the organelles you cannot see the all the things of the organelles, so because sometimes there are transparent materials as well. So in the light microscope it becomes difficult to see them. So the solution to this is, you add a dye on it. Like for example, you have taken the specimen, you will add a dye on it. In case of animal cell, or animal anything which you have taken from the animal, you will add methylene blue dye. Okay, you will add
Yeah.
methylene blue dye. And if you have taken the plant specimen, so you will add iodine solution. Okay,
Yeah.
that iodine solution will visualize all the things of that particular portion of the plant which you have taken for the examination. Okay, to check that under the microscope.
Yeah.
Okay, from here we use the light. Okay, from this side, this slide is used. The slide will focus on this region, and from the above side you see this. Okay, now they have mentioned a typical animal cell is about 1/40th of a millimeter in diameter. You see what they have mentioned, a typical animal cell is 1/40th of the millimeter in a diameter. Like for example, if you have one diameter, there is one diameter, and in that you have taken 1 millimeter, and in that 1 millimeter you add 1 millimeter by the 40, then you will get one cell.
Yeah.
So this you can imagine how small the cell is. This is rather a clumsy term, So scientists use smaller units like 1 meter contain 1000 millimeter. Okay, 1 meter contain 1000 millimeter, and 1 millimeter contain 1000 micrometer. So a typical animal cell is 25 micrometer. 25 micrometer. You know the
Micro.
micrometer, you know deci, centi, milli, micro, the prefixes.
Yeah.
Okay, so what is 25 micrometer means? 25 into 10 raised to
Like
power
into 9.
deci, centi, milli, micro. It is minus 6. 9 is nano. Okay,
Yeah,
micro is,
mine. Okay.
this is, you know, deci. Firstly we have, sorry, we have deci, then centi, milli, then micro. Okay, deci is 10 raised to power minus 1, centi is 10 raised to power minus 2, milli is 10 raised to power minus 3, and micro is 10 raised to power minus 6. Okay, so this is micro,
Yes.
10 raised to power minus 6. Okay, now are you clear to this? That the size, the
Yes.
typical size of animal cell is 25 micrometer. Mark this on your book. This is important information. Okay, now they have mentioned now how you can check the size of the structure of an organism. We will see this in the next lecture, because this is, the meeting is ending over here. I am sharing the new link. Okay,
Yeah.
this meeting is going to end.